exactly use optical tweezers to assess human red blood cell membrane stiffness. So now, before I move on, I would like to first tell you a little bit about the human red blood cell, just a small recap, let me say, because I think most of you are probably pretty familiar, and Professor Pomorski in the morning did give us a lot of insight about the human red blood cell. But we all know that its primary function is to transport oxygen to every part of the human body, all right? And the other interesting fact about it, it's probably the most abundant red blood, cell in the human body. In fact, 1/4 of the total number of cells of the human body are red blood cells. So at any given time, there are about 20 to 30 trillion red blood cells in the adult human body. Now, the shape of the red blood cell, the diameter is about between, let's say, 6.2 to 8.2 micrometers, and the thickness actually varies. There is a thicker region towards the edges, which we sometimes, we call the rim region, and then there is this biconcave, so there's this depressed part, which we call as the dimple region, and at the center of the dimple region, I think the thickness goes down to about a micrometer, right? And moving on. So this is, again, I think a lot of insight was given today morning, but nonetheless, let me just tell you what is really there in the cell membrane, okay? Now, the cell membrane, it has this lipid bilayer, and this is the example which Professor Pomorski told us about an asymmetric lipid bilayer. So essentially, you have an outer monolayer which has PC and SM, which we kind of heard of, oops, sorry about that, which we heard of in the morning, and the inner monolayer has essentially PE, PI and PS, and out of which PS was told to us was a charged lipid molecule, and it's never really seen in the outer layers, and when it is actually exposed is when the macrophages basically pick it off from the red blood stream. Now, along with this lipid bilayer, what is there in the red blood cell, which also helps it maintain its shape, is essentially this kind of a cytoskeleton, and it is made out of this spectrin proteins, and it forms a kind of a triangular network. Now, for all the purposes of this talk, what I am going to describe to you, we really don't distinguish between the lipid bilayer and the supporting cytoskeleton. We treat it as a whole, and the entire membrane stiffness that I'm going to be talking about is essentially for the entire structure, both the cytoskeleton as well as the lipid bilayer and all the, the proteins which are sitting there, transmembrane proteins which are sitting there, everything as a whole. Okay, so we don't, as it was said in the second talk today, this is coarse graining. Okay, so I'm going to do some kind of coarse graining over here. Now, so what do we know about the red blood cell? So the nice thing is, I mean, as from a physicist's point of view, okay, they are kind of less complicated. Why? Because they are anucleate. There's no nucleus inside it. And of course, lack of a nucleus implies that there is no DNA, right? Which means that red blood cells definitely have a birth and a death process, because they cannot do a cell division, correct? So the birth process is basically something which is called as erythropoiesis. It happens in the red bone marrow of large bones, and it actually is formed with a nucleus, and during the process of erythropoiesis, the nucleus is actually expelled, and the region is where it is expelled is basically where you get the dimple region, all right? And it also doesn't have other cellular organs such as mitochondria, Golgi apparatus, endoplasmic reticulum, and it also implies that not containing mitochondria, it cannot really use up any of the oxygen that it is transporting, okay? So this is certain interesting facts about the red blood cell. Now the point is, what did I say? I said that they transport oxygen. We all know it transports oxygen to all parts of the human body. Now they are transporting it through these blood vessels. Now the blood vessels in the human body can range from a few centimeters in diameter. I mean, like let's say a couple of 2 to 3 centimeters, I think, in diameter, down to about 5 micrometers, right? And the transport is something like this, a very, very simplified way which I am showing it. So it leaves the heart and the aorta, and then of course you go through arteries, arterioles, and finally you go through the capillary bed. So every red blood cell which is actually leaving the heart is finally entering the capillary bed, where it is encountering blood vessels which are about 5 micrometers, and I believe in the eye there are even blood vessels which are 3 micrometers across. And then it finally again these join back and go into larger blood vessels. Now what is the implication? If you remember, I told you that the diameter of the red blood cell was about close to 8 micrometers, let us say. Now the 8 micrometer object is essentially going to have to squeeze through a, let's say, a 5 micrometer capillary somewhere over here, which means it has to undergo a huge amount of deformation. So thereby the cell membrane stiffness, okay, the stiffness, the mechanical properties of the red blood cells are indeed extremely important, and they have a huge relevance to the way in which blood is being circulated in our body and the rate at which oxygen is reaching all our tissues. Now the point is that this still wouldn't be a big problem if the cell membrane stiffness was not being affected by certain common conditions that we have, okay, some disease conditions. So what I'm going to show you now is this is a whole bunch of disease conditions which you can have, all right, in which the red blood cell, the RBC membrane stiffness can get affected. Now what we have kind of tried to concentrate on in our labs is essentially on hyperglycemia, which we know is a very, or diabetes, okay, diabetes mellitus, which is a very, very commonplace condition, unfortunately, in India definitely, and perhaps even all over the world. So the passage rate of RBCs in narrow capillaries would be affected, which in turn would affect oxygen supply to cells and tissues. Now the question is, what we set out to find is that, okay, you know, first of all, can you really figure out what is cell membrane stiffness of, let's say, a red blood cell? And secondly, does it really get affected in the flow? I mean, does it affect the flow? So this is the premise on which we started our work. Now, of course, there are several methods. I kind of have mentioned this before in my starting slide in the earlier part of the talk. There are several methods to measure viscoelastic properties. Atomic force microscopy, for instance, and Chetna, who's with us, who has actually done work with red blood cells and AFM when she was doing her PhD. And then you have, of course, micropipette aspiration, stretching of an erythrocyte. I also showed you how you could use a, you know, kind of a sinusoidal disturbance to one end of an erythrocyte and measure, you know, in some sense, the stiffness or the mechanical properties of a red blood cell, magnetic twisting, cytometry, and so on and so forth. Now, the important thing to realize over here is the red blood cell is finally a viscoelastic material, let us say. Very simply, you know, I know I'm taking away from a lot of its beautiful complexity, but as a physicist, I will say it's a viscoelastic material. And very often the mechanical property I'm going to measure is going to depend upon the stress rate at which, I mean the stress range and rate which is being applied to it. So thereby, the properties that I come up with, okay, sometimes may be very different depending on the exact method that I am going to be using. So for instance, in the next slide, what has been done is, you know, you can see a cell stretching, all right, and here you have come up with the shear modulus about 3 to 6 into 10 to the power of minus 6 Newton per meter. And here the energy of deformation, this is all in some 10 to the power of minus 18 joules, is like something like 100 to 300, all right. Now this is of course much higher in something like micropipette aspiration. It goes from 250 to almost 750, and you can see that the range has completely changed of shear modulus, all right. Now the question is, what kind of viscoelastic mechanical property am I interested in? I am interested in putting forces on the red blood cell which it is going to see inside the human body while in circulation, because that is what is really relevant to us, because that is what is going to affect the passage rate of the red blood cells and the rate at which oxygen is going to reach the tissues. Okay? So I need to basically work at the same force regime, the same energy regime. So I have not yet talked about the first one is because I'm going to go into detail about this particular kind of measurement after this. So now that let us, so far we have talked about, like beads in optical traps. So now let us ask a question that I'm not taking something which is very highly asymmetric, but I'm taking a disc. So what would happen to a disc when I put it in an optical trap? Okay, so something like this. So there was this particular paper, okay, where they have worked on wax discs and also on PMMA discs. And what they found was that when they put it in an optical trap, essentially what happens to the disc is that it reorients. So if this is the beam propagation direction, basically the disc which is sitting flat would reorient so that its axis, okay, its diameter would be now sitting parallel to the beam propagation direction. So the question is, okay, one of the closest approximations to a red blood cell would be a disc, right? So would the red blood cell behave in exactly the same way in an optical tweezer? So this is basically, so human red blood cell plus an optical tweezer, okay, and let us see now what really happens. So this is essentially a series of photographs. They have been taken, I think, about 30 milliseconds apart. I forget the exact number, all right. Now these, as you can see, it starts off like this, flat, and then you can see that the final position is like this. So the question is that, is it like those completely rigid wax discs actually doing a reorientation, or is it folding? Because there were some papers, okay, some research work which claimed that it was folding. So now what I'm going to do in the next slide is essentially going to play for you a video of this particular, so you just watch what happens, you can see what happens. So it did look to us like that it was a reorientation. I'll just play it once again. It's a very short video. So you can see this is what the final orientation is. Now, when looking at the final orientation and the fact that it is extremely quick while it is, reorienting like this, people had doubts whether it was actually reorienting. So what I'm going to show over here is essentially the red blood cell has already reoriented, and now what is being done is that the laser is being blocked. So now, of course, the trap is gone. The moment you block the laser, the trap. So the red blood cell should actually go back to this position, or if it has folded, it should unfold, right? And this is going to be a little slower, so maybe we can observe it better. So let's have a look at this. So you can see this, the laser is right now on. Then it goes off, and you can see it seems like it is not unfolding, rather it is going back, so this is not a thing of unfolding, but rather going back to the original flat orientation. But then, yeah, so now the laser has been switched back on, and of course it's a very quick process. Now the thing is that we had a tough time convincing the people who we send it to. They didn't believe that it was reorienting, and the only way to convince them was to, when it was in this position, to, so this is a top view that you're seeing, right? You're looking from here, and it looks like this kind of an orientation. So the only way to convince them was to also look at it from the side. So this is precisely what has been done over here. So this is your optical tweezer, as you can see over here, all right? This is your tweezer, this is the sample plane, and this is your trapping objective, and this is your laser which is coming in, the trapping laser which is coming in. So along with that, another objective which was placed over here, and you could do a side view, all right, of this. And this is your side view. And of course, we didn't have such a fantastic magnification. This was at 100x magnification. This was much lower, but you can nonetheless make out that you can see the, oops, sorry. You can see the entire cross section of the red blood cell. So we were convinced that this was not a folding, but a reorientation. Okay, so now that you have, I'm hopefully everybody here is also convinced that it is a reorientation. It's a biased view, of course. It's me speaking. But nonetheless, what can one do with it? So one can basically, you have the red blood cell in the trap. It starts off at a flat orientation, and finally it is sitting at an edge-on orientation. So what you can do is you can basically measure the time it takes to go from the flat orientation to the edge-on orientation. Now what does this depend upon? It of course depends upon the red blood cell, but it will also depend upon the laser power, right? So larger the laser power, the larger it's going to be the torque, so the faster is going to be the reorientation. So if we did it for a rigid disk and we just kind of wrote up this kind of an equation, I am not very sure where that has come from. Okay, I'm sorry about that. So you have this kind of an equation written up for this, and you find that basically the reorientation time, and this is in seconds, and this is your laser power in watts, that as you increase the laser power, you expect the reorientation time to come down. So this is for a theoretical rigid body which has the same shape as the red blood cell, all right? However, when you actually do the experiment, so these are our position, the actual measured points, and this shows us the fluctuation, the error bar on these particular measurements. You find that it should have actually followed this black line, but instead you see that beyond a point it is following something else, okay? It is not really following the black line anymore, right? So obviously, now let us look at the whole thing. We are pumping in energy to make this turn happen, and if you see the rigid body, which has the exact same shape as the red blood cell, which is kind of derived from its behavior at low powers, predicted that it should take less amount of energy, because more time it sits, more energy it takes, right? So it should have taken less energy to do the reorientation, but instead of that, it is actually taking some little bit more time and thereby a little more energy to complete the reorientation process. Now what is the problem with this black curve? This assumes a perfectly rigid body, and we know that the red blood cell is not a rigid body, okay? So this difference that we are getting, okay, between the perfectly rigid body of the same shape and size and the actual measurements must have information about what is making it not a rigid body, thereby its cell membrane stiffness, its viscoelastic nature, whatever you want to call it, all right? So this must be, the information must be hidden in this difference. So what we did after that was essentially, this is your laser power, and this is basically the difference in the energy that we are seeing, okay? And initially it was, sorry about that. Yeah. So what you see is that it essentially goes through a maximum and then comes, okay, so let me just show you. At very high laser powers, we actually cannot measure, all right? We cannot measure, because what happens is that it will cause the red blood cell to get damaged. So what you do is that you can fit a straight line through these, okay, all these points, and then you extend it even to powers where you're not actually making the measurement, and then you come up with a graph like this, okay? So you can see that the extra energy increases and then goes through back to zero, right? There is a maximum energy that we get, and the question was that, can this be a marker of the cell membrane stiffness? So that's the question we asked. Now if you are using just red blood cells which have been from a person who has no chronic medical conditions, obviously you're not going to really figure out too much. You are going to get some delta Emax values and maybe they will match, but you're not going to get any further until you test with red blood cells which are faced different conditions. So what we did was that we actually got red blood cells from an individual who had been, it was confirmed that he was hyperglycemic, but nonetheless he was not under medication, okay? I use he generally, okay? But not under medication, which was a little important for us. So these are now something which we call as HRBCs. The other one, what was done was, I think it was described by Professor Pomorski today morning, that you change the conditions out of a red blood cell and they essentially go into this kind of a spiculed structure, all right? And how we did this was that by slightly altering the pH around the red blood cell, and then when you you kind of change the pH back to what it was meant to be earlier, all right, you essentially could take this particular structure into the red blood cell. But what it actually meant is that you had subjected the red blood cell to an osmotic shock and then restored it to look like the original, but however, the cell membrane still had signatures of having undergone that osmotic shock. So they looked like the normal red blood cells, but they had actually undergone an osmotic shock and come back. So these are what we call as restored red blood cells, so RRBCs. So now what is expected from seeing other literature is that these HRBCs are likely to be stiffer than normal RBCs, and these RBCs, the RRBCs, are likely to be more floppy, if I may use that word. Okay? So let us now see what we found. So this is essentially, so let me just put in the values. So what we are looking for is that maximum change in the energy, the delta E max as we called it. And we did normal RBCs 2 studies. You can see that this delta E max values are pretty much okay with each other. One is about 8.6 in 10 to the power minus 18 joules. The other is about 8. So it's not bad. When we did it for this hyperglycemic RBCs, we got something like 18.7. And the restored RBCs, I mean, it depended on the exact way in which we had done it. We got way lower than what we would get for the normal RBCs. So it kind of assured us that perhaps this was a good metric to use just to understand what the cell membrane stiffness was like. Now, however, this is of course in an optical tweezer, and do we have a different way of doing this experiment to assure us that this can be used further? So now I will tell you a slightly different story. The idea was that, people can tell you that, oh, you have taken the red blood cell from a person who has now a chronic case of diabetes, but you didn't measure his red blood cells membrane stiffness when he didn't have the problem, and perhaps it was always high. Okay, how do you know? So your delta E max must have been higher even then. So how do you go about, doing it? So what was thought of was that you take red blood cells from a single person. So now there is of course a small variation in the thing, but it is from a single source, a person with no known medical conditions. And then what you do is that you basically, in the suspension of the red blood cell, put in a certain concentration of bovine serum albumin, okay, so BSA. And what was found is that for different concentrations of BSA, the behavior in the optical tweezer actually changed, all right. So so you can see, I mean, so these are for certain, concentrations which it has been shown here. I have gone through these graphs before, so I'm going to move on from here. But the concentrations that were worked on were these, they have been just listed over here. Let me go on over here. And what was found is that depending on the concentration, so what has been given in these brackets is the concentration of BSA used in milligrams per milliliters. And you can see that about 0.5 of that is kind of giving us a close approximation of the untreated red blood cells, okay? And as you go higher than that, you can see that the numbers of this delta E max also keeps on increasing. So in some sense, you can take red blood cells from a single source, and then using different concentrations of BSA, you can tune the cell membrane stiffness. Now, of course, this is a very useful method because now we have red blood cells from a single source. We know that we have only tampered with the cell membrane stiffness, and now we can go ahead and try flow experiments with it. We can also do it, I mean, we have already done it in the tweezer, and finally see whether there is really any correlation with what we measured in the tweezer and what is coming out, let us say, from our experiments in the flow. So now, how do you do the flow experiments? Okay. So for this, we actually chose cyclopo filter papers. Now the reason why we chose cyclopo, I don't know whether it's a familiar concept with all of you, but for me it was something new, so I will just share it. I always thought filter papers were essentially, membranes through which there were these nice, channels which went through, which had a single diameter, very well characterized, straight, okay. but apparently not all filter papers are like that, which we found to our horror. And these were filter papers which actually work like that. So these are polycarbonate membranes in which actually it's track etched, in the sense a high energy particle is sent through, and then that region which has been damaged by the high energy particle can be etched out chemically. And you have precise control on the diameter that you actually get out of such kind of filter papers. So you can see this is a kind of a cross section of a filter paper. So there are these neat little channels that you get which are straight and practically uniform diameter. So we chose two kinds of filter paper. Now what was done was that one was about 7.92. I mean, this is actually, we measured all these things in the lab, and this 7.92 micrometers will tell you that it is just about the diameter of a human red blood cell. On the other hand, the other one, the 5 micrometer, is smaller. So now what does it tell us? That if the red blood cell has to travel through this particular filter paper, it does not need to undergo any kind of deformation. However, if it has to go through this 5 micrometer channel, obviously it has to undergo deformation. So now the whole idea is that let us see how much time it takes to go through these filter papers. And in our estimate, it should take a longer time to go through the 5 micrometer, simply because it has to actually deform, and then it will fit into the 5 micrometer, and then it will travel across. So that's precisely what we did. And this is a very, very interesting thing that we saw. So please note, what is on the x-axis is the concentration of BSA with which it has been treated. And what we saw from the tweezing experiments, that the higher the concentration of BSA, the larger the cell membrane stiffness, or at least our delta Emax values were definitely increasing. And these are basically the number that is passing through per minute at the moment. I think it is in 60 seconds. In the case of the 8 micrometer, what we found that as the cell membrane stiffness increased, the number that actually went through in a minute increased. And the reverse is true of the 5 micrometer pore. You can see that as you have increased the stiffness, the number is reducing. Frankly, we didn't expect this. Okay, this we kind of expected. This was new for us. So what we had to do after this was to figure out, okay, let's find out the total time of passage. Okay, so we said this is the total time of passage, and this can be divided as a time that the RBC requires to deform to get into the passage, and the other is the passage time itself. So basically how much time it takes to go from one end to the other. It is a simplistic approach, but it's the best we can do. Now, what was done in the case of the 8 micrometer pore, okay, since the deformation time is actually 0, the entire time taken for it to go from one end to the other is just the passage time. No need for deformation. So we in some sense knew the passage time, and we knew the length of the filter paper, the thickness of the filter paper it's going through. So we could calculate the velocity at which it is going through. So this is essentially the velocity at which the red blood cell is traveling through the 8 micrometer pores. And as you can see that with the increase in the cell membrane stiffness, the velocity seems to keep increasing. So then what we had to do is we had to figure out a kind of a translational drag from this. Now what we say is that the translational drag depends upon the red blood cell, and even when it is in a 5 micrometer pore, it must have exactly the same translational drag. Okay? So we estimate this beta. Okay, over here the table has been put. This is for the different concentrations of red blood cells, and this has been done essentially from the behavior in the 8 micrometer pore. We have got basically the translational drag, and you will notice that as the cell membrane stiffness increases, okay, with the BSA concentration, the drag actually reduces. That is kind of evident to us from the fact that it seems to be traveling more easily in the 8 micrometer pore. Now we of course have to use this in the 5 micrometer pore. In the 5 micrometer pore, we know the thickness, we know the shape, so we have assumed that it gets deformed into an axisymmetric phase. We know that there is going to be a certain velocity it is going to get from the translational drag and the shape factor, this is basically the estimated velocity from which we can get a time of passage. So if I go back here, we can estimate this Tp, and then T, the total time minus the passage time, will give us the deformation time. So this is the analysis that we have done and the estimate that we get. And you will notice that this deformation time that we get over here now, you can see that it goes from about 7.6 milliseconds for a normal RBC, because 0.5 we take is, you know, the closest to a normal RBC, and then 1.35, it has gone to more than thrice its value. All right, almost, yeah, more than thrice its value. Okay, 3.5 times. So now what we did was that, yeah, so this is basically what I have given you, the assumption with respect to the RBC shape once it is deformed. We have taken it to be an axisymmetric shape, and then using the Helfrich elastic energy model, I think today morning's lecture, Abhishek kind of mentioned that he's going to tell us about the Helfrich Hamiltonian. So using the Helfrich elastic energy model, we kind of get an energy deformation, which of course depends upon the elastic bending modulus of the red blood cell. Now using this, I will not go into too many details, we essentially get the bending modulus of the red blood cells as a function of the BSA concentration, and this is entirely gotten from the time that is required to deform. Now of course you can ask me the question, and that is very crucial over here, why is it taking time to deform? Because it of course requires energy, right? And where is that energy coming from? That energy is coming from the blood flow. Our heart is constantly pumping, and what is going to happen is that it basically requires that much time, because that much energy has to be incident on the red blood cell, and it's only when that much energy is incident it is going to be able to take on that shape. This is very, again, very simplistic. It's going to be a continuous process and not a sudden process, but nonetheless, this is the rate at which we can say that the blood flow is delivering energy to the red blood cell, and this energy of deformation which is required divided by this incident energy is going to basically give us the deformation time. So from this, essentially we have back calculated and gotten this bending modulus. So now this is how the bending modulus is changing with the BSA, with the stiffness, and of course as expected the bending modulus increases as the stiffness is increasing. Now the important question to ask is now, what is the correlation with our optical tweezer measurements? Okay, so is it really correlated or is it not? And this is what we got. Essentially this is your measurement from the tweezer, this is your measurement from the flow experiments, and you can see that the bending modulus and the delta E max factor which you could extract from your optical tweezer dynamics, you can see that there is a pretty good linear correlation. So in some sense this is like a calibration. So from your tweezer measurements you could get to the bending modulus or vice versa, right? Now, okay, the question that one can say is that, all right, I can see that BSA treated red blood cells are basically taking a longer time to move through these straight nice passages. However, in the human red blood circulatory system, this is not how our blood vessels are, right? So our blood vessels are more probably like this. So you should try doing the same experiments but using filter papers which resemble our circulatory system a little bit, more closely. So these are essentially cellulose acetate silver filter papers. This is not, let me just give you a warning that this work is not yet completely analyzed, because we ran into some kind of trouble, but nonetheless the result is a bit interesting, so that's why I'm sharing it with you. And what are these? These are essentially a stochastic network, I mean it's just been randomly put, and you can see that there are pores over here, all right? So you don't get basically the same pore size as you move on, so the network, it's like layers, but the same pore size need not necessarily be seen. However, they come with a characteristic particle retention size, which means that as the material percolates through or like moves through, I should not use percolate, as it moves through the filter paper, it is going to encounter, let us say, at least one pore which is smaller than the particle retention size. So this is the, the takeaway that we took. And so what is the result over here? Now the result over here, we got particle retention sizes of 3 micrometer, 4, 5, 6, 8 and 11. Now what you can see is that when the red blood cell, when the particle retention size is actually larger than the red blood cell size, the behavior is pretty much like what we saw in the single passage, in the sense that as the stiffness increases, you can see the passage rate has also increased. And the same holds when the size is smaller, which is that, with increasing stiffness, in some sense, the flow rates are coming down. But in between, something really strange has happened. And we have actually been trying to model it. And to model it, we need actually the pore size distribution of these filter papers, which is where we kind of ran into a roadblock, because we had to use a mercury porosimeter and there were not too many available. So what we are currently trying to do is we are trying to work with scanning electron micrographs of these filter papers, and then from the images trying to kind of get a distribution of the pore sizes. So once we do that, the model which is already ready can be put into effect, and maybe we'll be able to understand why there seems to be a kind of an optimum stiffness at which the flow rates seem to increase and it's not so good elsewhere. Okay, so this kind of a strange oscillation which you see maybe can be explained. All right, so now, yeah, so this is something you can do on the tweezer. And here basically, unlike watching just the dynamics, what has been done is that you kind of take a power spectral density of the trapped red blood cell itself, right? Now once you have done the power spectral density, you can always get a corner frequency, okay, associated with that. It's a pretty noisy spectrum. It's not like as nice as what you can get with beats, but nonetheless you can still, make a fairly okay fit and you can get these frequencies. Now what you will notice is that this is for the normal, this is for the normal red blood cell over here. And as you treat it with more, BSA, so which means the red blood cell membrane stiffness is increasing, you see that the corner frequency, okay, actually reduces, right? Which means that it looks like the red blood cell is being held in a it's being held less stiffly, let me put it that way, or the trap stiffness has somehow gotten lowered. And this is the case where you have treated it with a very little BSA, and actually what happens over here is that the red blood cell doesn't, it behaves like the restored red blood cells. So its membrane actually becomes floppier, and you can see over here that the corner frequency seems to be higher, which means more stiff trap, okay, in some sense. So the question is, what is really happening here? But before that, we tried to see some kind of a correlation. So one correlation, of course, the kind of behavior, you can see this is your BSA concentration, this is your corner frequency, and as the BSA concentration increases, which means the cell membrane stiffness is increasing, you can see that the corner frequency actually starts falling. And this is basically the corner frequency measurements and the delta E max, and you can see it looks like an inverse relation that we are getting. I have not yet put the error bars and all, so these are kind of not very strongly analyzed results yet. We have the data in place, but nonetheless you can see that it looks like an inverse relation. So the question is, what is really happening over here? And so this is our conjecture about what could be possibly happening. See, RBCs are of course not perfectly rigid bodies. They are also extended bodies. Now when you are trapping and you are trying to take a power spectral density, you are obviously measuring fluctuations, correct? So you are seeing how the RBC is moving around in time. Now the question is, since it's an extended body, you are probably monitoring only a part of the RBC, maybe the central region of the RBC. Now the RBC, of course, is being subjected to hits on all sides, right? Now the question is, how well is that particular, let's say, Brownian, motion, which is basically started off at one end of the RBC, coming through to the region which you are monitoring. If it is a perfectly rigid body, you know it will be perfectly reflected. However, if it is the elasticity in between the point of impact and the point where you are monitoring is different, then you may not get the entire information. So, the more stiff it is, the more well it kind of moves as a coordinated system, and thereby it appears that the trap is less stiff. However, if it is more floppy, maybe it is possible that that fluctuation is not even getting registered here. So it looks like the body is held in a much stiffer trap. So thereby, we tried then, seeing whether the inverse relation was indeed true. And you can see this is the corner frequency. This is the delta E max, the factor which we got from the reorientation experiments inverse. And again, you can get a perfectly straight correlation. And this we tried in 2 different rounds with using 2 systems to see we were indeed, on the right track. And you can see a good correlation, a linear correlation in both times. So, finally, what can I say with the optical tweezer? You can get a measure of the cell membrane stiffness, it appears to us, both from the reorientation dynamics as well as from the power spectral density. What you can, from the reorientation dynamics, you can come up with this particular factor, the maximum energy required for reorientation, and that seems to be something that scales with the cell membrane stiffness. Now, what we have done is that we have, of course, correlated it with the flow rate of these red blood cells through these cyclopore filter papers, which are uniform diameter pore filter papers. And what we find is that as the red blood cells, their cell membrane stiffness increases, deformation time increases, and of course from this we have calculated their bending modulus. The bending modulus also increases. Now, we have also found that the corner frequency can be a measure. Another thing that can be a measure of the stiffness is purely from the flow measurements. We found that as the cell membrane stiffness increases in larger diameter pores, it looked like the stiffer red blood cells could travel faster. So that particular translational drag parameter could also be used as an assessment of red RBC cell membrane stiffness. So I think we still need to do a lot of calibration work to ensure that these are indeed good and whether they can be even taken on to the next level as a diagnostic tool. But that is something that the future will hold. So I would like to thank all the members of the optical tweezer group at Bangalore University. Professor Sharath Ananthamurthy actually built the tweezer facilities. He's the first person who started off all the experiments. But since then he has moved on to University of Hyderabad and is currently Vice Chancellor of Kuvempu University, so he's no longer with the group. Sahana is a current student. She's working on the same problem. And these are all people who actually worked very strongly with red blood cells. Chetna actually didn't do red blood cells on the optical tweezer. She did avian red blood cells, and she worked a lot with the atomic force microscope. These are people who actually worked on the red blood cell, Rekha, Praveen and Yogesh. And these are students who did some other experiments. And I would like to thank all these funding sources. And of course, this is a view of our university. This is the physics department, and I would be very happy if you ever come down to Bangalore, please visit us, and I would be very happy to take you around the lab. And I'm open for questions. Thank you all for your attention. Any questions, please? Yeah. This was a very technical session, I think. Yes, I mean, definitely. I mean, that was actually the idea with which we started. The reason why we went for hyperglycemia is because it's a very common problem, all right? And my idea was that, a lot of the symptoms that you associate with diabetes, okay, could it be actually related to impaired blood flow? I mean, it's a very, very simplistic kind of drawing of lines, but that was the idea with which we kind of started off. But yes, the hope is to kind of make this a feasible way of studying RBC stiffness. So for instance, one of the ideas we had was that COVID-19 is supposed to also alter RBC membrane stiffness. Okay, so I had read some articles on it, and we were hoping that, maybe we can work with some, stored. Unfortunately, it's not a very, it's a very sensitive technique, and you need to basically work on the red blood cells within 4 hours of extraction. So we have to figure out how good it will be as a diagnostic tool. But yes, the final goal is to end there. And of course, you can ask the question, will it, confirm that somebody has a particular disease or not? Maybe this is not the best way. But if there are complications arising out of the disease, one can see whether the membrane stiffness alteration has also something to do with it. Okay? And then, or if the membrane stiffness, the sign appears before other markers for the disease. So whether, you can do an early detection. So that is the reason, that is where this will come in. Because it's not a trivial method to do. That I completely agree with. It's not trivial, and you have to do it within 4 hours of the red blood cell being extracted. Storage conditions have to be really very stringent. So there are all sorts of things, because everything affects the, the measurement, the dynamics which you see in the tweezer, everything affects it. But nonetheless, I think there are certain niche places where it can become more useful. Yeah, so that's the hope.